Good morning, everyone. I'm Christopher Clemmensen, the group leader here at CBMR, and it's my real pleasure to kick off the meeting and to introduce the first speaker, which is President Matthias Tschöp from the, I mean, that is your title, from the Ludwig Maximilian University in Munich. I really want to try and make it short, but it's difficult because Matthias has so many achievements that I think is exciting to highlight, right? So he's a German physician that at one point took a kind of a research stint with Eli Lilly in the US. And at that time, he published the first paper showing that ghrelin is a food intake promoting hormone, published in Nature, cited more than 5,000 times, worked a few years on ghrelin, and then decided to move on to work on what we today everyone knows as these multimodal peptide hormones that act at several receptors. So more than 20 years ago, Matthias, together with the chemist Richard DeMarchi, developed the first version of these molecules, preclinical studies to demonstrate their efficacy in rodents. So, extremely pioneering work. And that was when he was at the University of Cincinnati, and then he was recruited back to Germany in 2011 to start at the Helmholtz Institution in Munich to start the Institute for Diabetes and Obesity, and that he quickly grew out of that to start the Helmholtz Diabetes Center, and then he decided to start the journal Molecular Metabolism that we all love, and the Helmholtz Pioneer Center, kind of moving within the innovation space at the time, and then finally he was promoted to be CEO also of the Helmholtz and take upon the role of the leadership there. And then more recently took up at the new position at the LMU as president. He's won a gazillion prizes for his work, mostly in the dual and triple acting peptides. And what else to mention? I don't know. I think he's a pretty nice guy, and I think it's safe to say I spent I spent a few years in Matthias' lab as postdoc, and he is actually a nice guy, as Julien says. And I think there's even though he's been very busy, we had many random evenings where we went out to play soccer, went to the cinema. So he's a kind of, I don't know how you make it work with 24 hours a day. But we're really happy to have you here, and I'm actually very excited to learn about your newest achievements and discoveries in this space. Matthias, welcome.
Thank you so much, Christopher. Some of what he said was true. I actually did bring some new data for those of you who have heard me speak in the recent years. We have a little bit of new stuff that will be at the end. Julien has already heard it, and I think she approved. So 3 weeks later, you get to hear it again. Well, let me start by introducing my friend and colleague Richard DeMarchi, who I have been working with closely over the last 23, almost 24 years now. He's a chemist, fantastic peptide chemist, and everything you see today wouldn't have been possible without our collaboration. We started out decades ago with a clear vision and goal in mind that we want to come up with new drug candidates for obesity that could be superior in action, more benefits, fewer side effects than what was out there at the time. And at the time, there really wasn't much out there. So we said, well, probably you need multiple receptor targeting. One signal alone, as we learned from leptin and ghrelin and so on, is likely not going to do it. Also, we wanted to base our therapies on gut-brain signaling, thinking that, well, getting things from the outside to the right places in the brain just hasn't happened. We don't have the technology. We want to use Mother Nature's toolkit of going from the gut signals that know where to go to influence metabolism in the brain. And then maybe find ways to have single molecules, and Richard had some ideas how that could be done, putting these different signals in a single molecule. Now, we started with this peptide family that comes as no surprise to anybody in this room, I'm sure. But at the time, it was not looked upon as something obvious, even though GLP-1 had started to emerge as an agent that can influence appetite and then body weight, but glucagon and GIP were not thought to be likely to make a beneficial contribution to an anti-obesity therapeutic. So we actually looked at glucagon first, and one of the major assets in the lab was that Richard could make molecules that were soluble, longer acting, and more powerful, so you could study glucagon action in a way that previously was difficult to do, with new agonists that had the required characteristics. Now, making these molecules depends on chemistry, and that chemistry has really evolved. I think that's one of the major success stories of this field, how chemistry had evolved to turn very short-lived endogenous hormones into longer-acting peptides. One of the things that I want to mention here is the helimonster Heloderma horridum, that has a saliva in which there is a version of GLP-1 that is longer acting, and that became sort of one of the steps forward toward actual drugs. The so-called CX tail at the end of the molecule was something that Richard also used. We used to put on glucagon and make that more long acting. And I'm telling that story also because about a year ago we had a nice alumni meeting with Christopher and everybody, and my former postdocs and grad students gave me a gift, and that is my personal buddy, which was called Matt Jr. And sometimes now he's not living with me; he's in a zoo. But sometimes I can go pet him and hang out, and it's good to have a friend. All right, so glucagon receptor agonism we then showed, and this was a special eureka moment in 2006, that if you make a longer acting, stable, potent glucagon and you put it in diet-induced obese mice, you can decrease body weight powerfully, comparable to what is possible with a long-acting GLP-1 receptor agonist. And we thought, well, two very different systems, two powerful actions, that could be the grounds for a first dual agonist. That was what we wanted to do. So, but that is discovery one, that glucagon receptor agonism can be helpful and put into therapies for the treatment of obesity. Now, for the dual agonists, this took a couple of years because the idea wasn't to sort of stick two of them together, but engineer one of these peptides with a few amino acid exchanges so that both receptors in the right balance could be activated. That then succeeded, and that has been published, and all of you, probably many of you, are aware of these data. You can decrease body weight dose-dependently with these dual receptor agonists, glucagon and GLP-1 receptors. And luckily, and to some surprisingly, we didn't do any bad stuff to glucose metabolism. Insulin decreased, blood glucose tolerance improved. So a glucagon agonist added to a GLP-1 receptor agonist did great things, which was published and translated into the clinic. There is a number of versions out there, phase 3 trials now. This is data out of Boehringer Ingelheim with their version, survodutide, phase 2 trials showing that this works in humans. Now, discovery 2, dual agonism, glucagon receptor, GLP-1 receptor, dual agonism, a single molecule, outperforms mono-agonism, at least preclinically, and I think in the clinic shows promising data, and approval will likely happen soon. Now, the next one we were focusing on out of that family is GIP. Now, that one was even less likely to work, many of our colleagues thought. But again, if you make longer-acting GIP molecules that are potent and activate the receptor specifically, you can see that there is impact on body weight in diet-induced obese rodent models. So just to go quickly through how that is done, you make a number of these agents. You got to make sure that not only they're stable, they are working at the right receptor, they're specific, they're potent, but also they work in mice and humans in both systems, because otherwise translation becomes tricky. First thing, do our GIP agonists decrease blood glucose? That's what they should do as an incretin. Yes, they did. They did decrease body weight, which was promising to us for potential synergy with another agent like GLP-1, for example. And then we wanted to see if they're specific. So we needed to show they're not working anymore if you knock out the GIP receptor, but they're still working if you knock out the GLP-1 receptor, and that was what we could show. Now, today we know that GIP receptor agonism also leads to body weight loss in humans. There's a number of pieces of data out there. There's a paper out by Eli Lilly with their version of a GIP receptor agonist. Now, these effects aren't as powerful as some of the other agents that we know of today, but certainly we thought this was an interesting addition that we could take advantage of. So next discovery was GIP receptor agonism, and as we still believe, not antagonism, offers significant therapeutic potential for the treatment of obesity. So next, dual agonism, GIP, GLP-1 receptor co-agonism. Again, took 2 years, I believe, until we had molecules that seemed to work in the right balance. We could show preclinically that they dose-dependently decrease body weight pretty impressively. And that was published now, 15 years ago or something, 13 years ago. And we were the first to put that also in the clinic for our first clinical trial together with Novo Nordisk and Roche, showing that this actually has beneficial effects in humans in terms of weight loss and handling glucose. Now, we all know GIP, GLP-1 receptor dual agonists have changed our world, at least the world of clinical obesity treatment, with the version that Eli Lilly made with tirzepatide, a GIP GLP-1 receptor dual agonist model after that example. And with that, you can find an average of 23% weight loss. So when I started as a young physician in the early 90s, that was thought to be impossible, that you can come up with a treatment that is safe and can reduce body weight in humans by an average of 23%. Now, with all the excitement that now is possible, sometimes we tend to forget that this is also a fantastic diabetes drug where about half of the patients treated that have type 2 diabetes can normalize their HbA1c. Now, an interesting aspect of tirzepatide is that it has a 4:1, 5:1 GIP GLP-1 ratio, so there's a lot of GIP in there, and it has a little bit of a different therapeutic window as, for example, GLP-1 receptor mono-agonists, meaning, yeah, there are also side effects, emesis, nausea, gastrointestinal effects, etc. But relative to the benefits to the weight loss, that is less. And it may have to do with the fact that the GIP receptor agonist doesn't cause the same side effects as a GLP-1 receptor agonist. And that may even mean that GIP receptor agonism counters some of the GLP-1 receptor effects. As you see on the right side, this is work by Matthias showing in the right model, the musk shrew, that they show emesis when you give them GLP-1. They don't do that when you give them GIP. And if you give them both, they're also showing less of the effect. Now, that brings me for a moment to the topic of would GIP receptor antagonism be better than GIP receptor agonism? Because there's this conundrum that both ways lead to decrease in body weight, which seems difficult to understand at first. We believe this has to do with the fact that both are very different based on the mechanism how they achieve that weight loss. So when you look at single-cell sequencing of the targets in the hindbrain of GIP receptor antagonism and GLP-1 receptor agonism, you see that they're very, very similar. On the other hand, however, GIP receptor agonism looks much more like GIP-GLP co-agonism, like the dual agonist. So what we believe is happening that indeed GIP receptor antagonism does mainly one thing. It sensitizes to and enhances GLP-1 receptor action. So what you do is a super GLP effect, and that leads to more weight loss. And GIP receptor agonism is real synergy between two different pathways. Now, that in our mind means that if you enhance the benefits of GLP-1 to an absolute maximum, you might also enhance the side effects, the adverse effects. I mean, you take a look at the recent New England Journal paper on the GIP receptor antagonist, GLP-1 receptor agonist maridebart cafraglutide out of Amgen, and you take a look at the side effects, adverse effects reported there. Indeed, you have vomiting in basically up to 100% of the patients, which seems to be in line with what we mechanistically uncovered. All right, so we think dual agonism, and that's the next discovery of GIP receptor and GLP-1 receptor activation outperforms monoagonism and is the better approach as compared to mixed agonism antagonism. Now, if you have two dual receptor agonists that work well, of course, it occurred to us early that there could be a triple receptor agonist. So how about a GIP receptor, GLP-1 receptor, glucagon receptor, triple single molecule co-agonist? That took much more time. There was, I think, 3-4 years of work going back and forth because the balance becomes more tricky, the chemistry becomes more tricky. You really have to figure out which amino acid in which position is the right one. It's sort of like playing with a Rubik's Cube, only the one switch of the cube means you're driving from Cincinnati to Indianapolis and you're making molecules and you're putting them in cells and purifying them with columns and putting them in mice and putting them in rats. Oh, it's the wrong one, and go back. But so it looks like this, and luckily many of or several of the positions in the molecule are shared by glucagon, GIP, and GLP, or at least two of them. So there it becomes a little bit easier. And then you do the enhancement of fatty acids that help albumin binding, of a CEX tail, etc., DPP4 cleavage protection. Now, in the end, we succeeded with the first triple receptor agonist that was superior to the dual agonist in terms of reducing body weight, reducing food intake. When you look at these curves, the dark purple one is a dual agonist, more similar to tirzepatide, and the orange one, that is the first triagonist, more similar to retatrutide. Now, that then held true for fat mass and for cholesterol. That was true for blood sugar levels and insulin levels, and it cleared the liver of fat impressively, which may have to do with the fact that now you have glucagon receptor in addition as a target as compared to tirzepatide. So you really have the liver as a target because that's where you find glucagon receptors, but not really substantial amounts of GLP or GIP receptors. Now, that also got published more than 10 years ago, and we're continuing to work and better understand these triple receptor agonist effects. For example, we made a mouse that, using CRISPR-Cas approaches, that doesn't have any of the three receptors. Turns out that mouse is alive and lives and eats. For us, it's important that we could show if you give that triple receptor agonist to a mouse that has none of the three receptors, nothing happens to metabolism and body weight. So we are as specific as we hope to be in our approach. So next discovery, a triple receptor agonist targeting all three receptors in the right balance outperforms dual agonists and mono receptor agonists. Which brings me to the summary of sort of the old data, saying glucagon agonism drives body weight loss and can improve metabolic health. GIP receptor agonism drives weight loss and can improve metabolic health. And we now have achieved, well, when you look at retatrutide, which has been out there for a while now in phase 3 trials with Eli Lilly and will be approved sometime soon, 30% weight loss. That is what has recently been published, that a triple receptor agonist can do 29%. So what impact has that made on the field, on clinical medicine? Well, we can already see that even epidemiologically. Obesity is showing signs of decline in the US. Now, I'm not saying that those are our drugs, but they certainly made a contribution, and this is what these new obesity drugs are doing. So there is impact, but what we're not there yet. There's a number of things that remain as challenges. Let me point out a few to you. One of them, the comorbidities that come with obesity, they're not reduced as impressively as we would hope when you reduce body mass index. So when you go from a body mass index of 35 down to 27, there's still a lot of patients that still have a lot of comorbidities of metabolic kind. So how do we address that, for example? Now, we started a company 2 years ago, Blue Water Bioscience, together with Richard and Timo Müller and Diego Perez-Tilve and others. where we wanted to build on our previous discoveries that you can also put small molecules, steroids, on peptides, and then that way have polyagonism and have the additional advantage of delivering these small molecules only to certain cell types and certain organs. And with that, you have a larger toolkit where you can address things by adding an estrogen effect, adding a thyroid hormone effect, adding a glucocorticoid effect, and maybe specifically address some of the comorbidities that are out there. The second point, some patients, for some patients, retatrutide might be too much, as we see already in first data, but for some patients, you need even greater body weight normalization. You need to have 40% weight loss. And how do you achieve that? Or how about patients where the available drugs don't work as well yet? So can we do even more? Now, when you look at the evolution going from mono to dual to triple agonist, there was always more body weight loss, down to 29%. But now Timo has come up with a way to maybe go beyond that. A quintuple co-agonist that is a incretin-mediated delivery of PPAR alpha gamma delta triple agonist with lanifibranor. And with that, he was able to show that when you put that on a molecule and you choose GLP-1, there's a little bit of body weight loss. But when you put it on a GLP-GIP, a dual agonist, then you have way more impressive weight loss. To be honest, as it goes in science sometimes, that surprised us. So there's something about GIP transporting a triple PPAR into certain cells that is a game changer for body weight loss. And as it turns out, for food intake and also for glucose handling. Now, if you make the experiment more interesting by comparing it to co-administration of a tirzepatide GLP-GIP and lanifibranor, you don't get that same effect. You have to have it physically connected so that lanifibranor is delivered only to those cells and not systemically available. And it's probably the cells that have GIP receptors, because you can't do it with GLP-1. This massively decreases fat mass, and it doesn't seem to have greater impact on lean mass. Now, when you compare it to semaglutide, also the new compound, the quintuple agonist, outperforms on every endpoint. Now, this is a paper that's now, I think, accepted and in press at Nature and will come out in a couple of weeks. Now, the third point is we still have issues with tolerability and with patients staying on the drug. As we all know, there's more or less complicated ways of dosing up so that patients tolerate these drugs better. Some patients don't tolerate it, some patients stop, etc. So can we find a GI gastrointestinal side effect-free version of a polyagonist is another question we pursued at Blue Water Bioscience. And herewith I come to our new data, which might or might not be a paradigm shift for incretin therapy. As you can tell, we are enthusiastic about it. And here's a provocative and symbolic way to look at this that got us into that direction. When you look at the history of these drugs fighting obesity now and being available to patients or being available soon, well, with semaglutide, you get, 17%, more or less. I mean, it's not about the exact numbers, it's about the perspective. So Vodutide may be able to do a little bit more when you add glucagon. Tirzepatide does significantly more, and retatrutide seems to be doing more as well. So you can look at this in a way and say, well, the less GLP we have here, the better these drugs work. Daring to say that here, but let's just bear with me. So this is the way we look at these drugs: mono-agonists, dual agonists, triple agonists, GLP-1 drugs. Well, if you really look at the contribution of how much GLP is in these drugs, that is not the way the chemistry really is reflecting this. Also, we have these complicated ways of dosing up, and that seems to be a consequence of the GLP-1 component, because when you now look at a triple agonist and for a moment think about where does the nausea come from, where do the risk factors come from, that seems to be mostly associated with GLP-1. So what if we have a GIP glucagon with a bunch of GIP and see what's happening? Well, frankly, it took me 4 years to convince Richard even to do that. And in the end, we pursued it, and it turns out that there's something there. So let me start with an experiment where we used retatrutide, the triple receptor agonist, put it into DIO mice, and there's a dose-dependent decrease in body weight. Now, if you put it in GLP-1 receptor knockout mice, you put a triple agonist in a mouse that doesn't have GLP-1 receptors, and now you still have very impressive weight loss. So this is now based only on the GIP and glucagon component in the triple receptor agonist. So this could be interesting. Now, today we know, as I mentioned earlier, that GIP agonism that was originally deemed not to have therapeutic benefit has therapeutic benefit. And then there are Lilly and other studies that show, well, in humans, this is also relevant. There's improved weight loss and glucose management with GIP. With fewer side effects in humans. That would confirm the hypothesis that maybe getting rid of GLP will help us get rid of side effects. So what we did was we put glucagon plus GIP in diet-induced obese mouse models, and we were able to show that that gives us really impressive benefits. So this is now a mix. You give GIP, you give glucagon, and you change the doses. You dose one up, you dose the other up, and you outperform even tirzepatide with that, which is interesting. Well, obviously there's a question of dose and so on. It's tricky. But you also improve glucose tolerance. It's not that you drive glucose through the roof, because apparently GIP does the job that GLP does in tirzepatide in buffering for what glucagon does. So we would hopefully be able to position a single molecule dual agonist, that's GIP glucagon, somewhere where nobody has gone yet, and have certain goals, once weekly, commercially viable dose, and competitive weight loss, etc. It may not have to be giving us more weight loss than retatrutide or tirzepatide, because having something that gives you significant weight loss and has much less side effects, adverse effects, maybe that's something we want to pursue. Okay, here it is. We made several GIP glucagon co-agonist single molecules that are GLP-1 free. And we can show that they are performing like retatrutide in preclinical models on percent weight loss, on daily food intake, on glucose tolerance, etc. That seems to be based on overall a decrease in food intake, but also a increase in energy expenditure, as pair feeding studies are indicating. We could translate that into another species. This also works in rats. And we have done non-human primate studies in monkeys showing that there's no evidence of GI intolerance in these monkey studies with a GIP glucagon dual agonist as a single molecule. So in summary, we believe it's worth to explore clinically if these new GLP-1-free dual GIP glucagon co-agonists indeed offer powerful anti-obesity effects without signs of toxicity and gastrointestinal side effects. That would add something that we haven't had before, but it could also give us additional flexibility in treating patients, tailoring treatments, because, well, at some point you could add a GLP-1 as semaglutide, and then you have a triple agonist. And then you can take it away. So you can tailor treatment over time to patient subpopulations. And we can even at some point then add, small molecules, steroids that can enhance what these dual agonists do. So do these patients need additional GLP? Do these patients need a PPAR? Or is a dual GIP glucagon sufficient for the patient population you're targeting? with that, I hope, I'm pretty sure I've given stuff for discussion. And I want to thank my close colleagues, Richard, Timo, Brian, and Diego. Everybody has contributed over time, including Christopher. And here's the slogan I had at Keystone. That is a little cheesy, but how about try hard or live GLP-1 free? I'm a die-hard fan, so whatever. Thank you very much.
Okay, thank you, Matthias, for that very, very interesting talk and also some provocative data, especially how dare you to come to Copenhagen and talk about non-GLP-1 based therapies.
Nobody's throwing stuff at
now
me
the
yet.
floor is open for some aggressive comments. Steve?
As always, Matthias, very excellent, stimulating, stylish, fascinating. So we don't have natural experiments where humans excessively make GLP-1 or GIP, but we do have natural experiments where people make too much glucagon. And they get odd things. They get suppression of their amino acid profiles, and many of them get skin rashes. How do you dial down the glucagon activity so you avoid what we know is the adverse consequences of chronic excesses of glucagon?
So I think the dose makes the toxin is specifically important for glucagon. And now there's now lots of data out of the dual agonism glucagon GLP-1 in humans, and it seems that the two things to worry about are obviously glucose metabolism, and the other one cardiovascular systems. We have some first data on both, and I can't talk about them yet, but all of the other things that you would see in glucagonoma seem to be at a level where we're not reaching into with our dosing. And having something else on board is of course important. So whatever of these toxic potential effects, GIP and or GLP are taken away is difficult to figure out. But apparently there's something that works. There's a path.
More questions? Don't be shy. Yes? Stretching? No?
What?
So very interesting and fascinating. I obviously have some questions because I work from the other side. So it's I'm for GIP antagonism, right? So you say that two questions. The first of, you say that GIP agonism works as a sort of as a supplement to GLP-1 activation, whereas GIP antagonism works only as a sort of a GLP-1 sensitizer. We have just put out data that if you combine a GIP antagonist with amylin, you see exactly the same thing. So it's not limited, or that's in monkeys, obese monkeys.
Yeah, but
so
you would enhance endogenous GLP-1 with that, right?
No, you just give you give amylin and you give a GIP antagonist, and you actually see almost synergism on the weight loss in the monkeys.
But you would enhance endogenous GLP-1 agonism as you sensitize the GLP-1 receptor. That could explain it.
So that could explain it. Yeah, that's right. The other thing is for the GIP glucagon thing. What about those patients that are actually diabetic, where we know that GIP has less of an effect? They're almost insensitive when you get an HbA1c 1/10. Wouldn't you leave those patients with just the glucagon effect, meaning that they will be overtly diabetic?
Absolutely. So our preclinical data indicate that may not be the case, but of course the clinic will then give us the truth. We have to figure out to which extent glucagon is unlocking something in GIP that GIP alone can't do, similar to what we maybe see in tirzepatide. I mean, these are philosophical discussions because you can't really mechanistically figure that out in the clinic. And the second perspective is, well, this could be a drug that is for obesity in patients that are not diabetic. And if you give it to patients that are diabetic, you may want to combine it with a semaglutide by a co-administration. These are two paths forward that we need to find out if they work.
Is there a question up here?
Thank you. Fiona McMurray, Novo Nordisk. Thank you. Really great introduction to this topic. And of course, the pharmacology is also really important. And you show the nice PK data at the end in your non-human primate study. I just wondered, do you also see efficacy in that study with body weight loss?
We didn't, because that study wasn't designed for that. It wasn't obesity models, but we're on that. Can't share data yet. Maybe one piece of data that I haven't been able to show today or haven't included in my data set regarding the antagonism and regarding Mats's question. If you give a GIP receptor antagonist to diet-induced obese mice that have no GLP-1 receptor, there's no effect. So that, I think, is a very clear piece of data that what you're doing with GIP receptor antagonism has less to do with the GIP pathway and more with the GLP-1 pathway.
Question up here.
Rodrigo Cataldo from Lund University. I was just curious, I think maybe I missed, but I guess you didn't show that. Regarding the last concept of this dual GLP-1-free co-agonists, glucagon GIP receptor agonists, have you measured the endogenous GLP-1? Is it rising as well? Or?
We haven't. Interesting question. Maybe that explains it all. I don't. We'll do that. I don't think we did. Good point.
There's a question down here and then up here afterwards.
Hi, Damien Demosé, Novo Nordisk. I was wondering if you ever look at beta cell mass or beta cell function preservation with all those combos. I mean, it could be interesting for long-term effect of those compounds.
Absolutely. We didn't do that with all of our compounds. We had one specific project where we made a GLP-1 estrogen dual agonist. And there we really focused on the pancreas and on the islets and could actually show there is significant preservation. Maybe even rejuvenation of the pancreas with that. We did a lot of single cell sequencing, etc. in that. There are 2 papers out, later one in Nature Metabolism, looking at exactly that. But that came out of our hypothesis that estrogen is known to have a preservative protective effect there. And these are very involved, expensive studies that we haven't done for the dozens of other compounds, but that would be interesting for sure. In my simple mind as a neuroendocrinologist, most of what we do with these dual and triple agonists happens in the brain. And we're focusing very much on obesity as a target.
There's a question over here.
Hi, thank you for the talk. Konstantinos Kokonos from Bioinnovation Institute. I just wanted to ask if I understood well from your dual agonist. From what I saw, there was no lean mass lost. So I was wondering if I understood well, the main mechanism of action was through appetite suppression. So could you speculate on why did you not see any lean mass lost?
Well, so first of all, there was not just appetite suppression. Actually, with that compound, we did see, because of the pair feeding studies, that there's something beyond that, which might be energy expenditure. And then the question is how much of that translates into humans. The data I've show that there is no additional lean mass loss compared to mono-agonism like a GLP-1 receptor agonist. Right? So this isn't the miracle drug yet that also makes you grow muscle or protects all of the muscle mass. In my simple mind, whenever you go on negative energy balance, you'll lose weight and you lose in a certain ratio fat mass and lean mass. And if that needs to be overcome, which is an interesting proposition, then we probably do need specific targeting of muscle, and that probably myostatin, etc. We'll need to find out if that is safe and how that can be achieved safely. With any of our dual agonists so far, I'll say that you only achieve what you achieve with the approved drugs. That is significant weight loss. Hopefully fewer adverse effects in the sense of gastrointestinal side effects, but the lean mass loss is the lean mass loss because of the negative energy balance that you're causing.
There's a question back here.
Yeah, hi. Thank you for a very nice talk. I really enjoyed this. And my question is very much related, I think, to this. So my question is whether you think there is an upper limit to how much weight loss you can achieve by mainly targeting appetite. Do you think we can just keep targeting appetite and then go lower and lower in weight, or do we need to combine it with other targets in order to have effective treatments?
Well, so the physiological answer probably is if you stop eating, you go lower and lower in weight, and there really isn't a limit. The question is what is healthy and what is medically useful. And I think that will more and more depend on the patient subpopulation you're treating, right? So if you have a quintuple agonist that gives you 40% weight loss, just speculating here what may happen in the clinic, then that is just for those patients who need exactly that. And there are those patients. And in the end, it's also a question of dosing, right? So there are reports that, oh my God, retatrutide in the clinical phase 3 trials has given patients too much weight loss too fast. Well, that's very simple. You can adjust the dose, right? And when you have a triple receptor agonist that is that effective and you dose down to 20% of the dose that is the maximally tolerable dose, then of course you also have much fewer side effects. And then that's a good thing. So having potent drugs isn't a bad thing, but using them responsibly with lots of expertise and with personalized metabolic medicine in mind is the key.
We'll take more questions. We have one up.
Thank you so much. Great talk. Herman Carrasquilla, Novo Nordisk. Over here. Here.
There. Okay.
So my question is in connection to adding hormones, corticosteroids or estrogens or any other hormone. You mentioned that. So I'm just trying to understand, how do you see this? Do you see as an improving efficacy or reducing adverse effects? Can you elaborate on that?
Both, actually. So that is the fascinating thing with adding these small molecules, because you're not only expanding the toolkit, now you have way more possibilities because you have way more hormones. You also have enormous power. I mean, these nuclear hormones, thyroid hormone would be fantastic obesity drugs if it wouldn't do a couple of things you don't want. Glucocorticoids are a fantastic anti-inflammatory agent, but there's some side effects there. So, but what if you could deliver it to only those cells where they do the things you want, and keep them away from the cells where stuff is happening you don't want? And we believe that is possible with the right shuttle. And in addition to having a peptide shuttle that achieves that, there's synergy between that peptide shuttle and that hormone. So, this will require a lot of in-depth investigation, because before you put an estrogen in a clinical trial for obesity, before you put a thyroid hormone in a clinical trial for metabolic disease, you better be sure that you minimize the risk for side effects. But I strongly believe this could be a completely new dimension of personalized metabolic medicine if we can do that safely.
And another one, if I'm allowed, it's going to be quick. So when you were showing, because sometimes there's so much focus on the percentage of weight loss, but then my question in the context of the different compounds, how the dosages are comparable, that's one question. And how good are we in moving forward from just BMI loss and focusing on losing fat or lean mass? And then with this I finish.
Well, the BMI discussion would probably fill another hour or so. But when we compare these drugs, we compare them mole for mole, number of molecules. These peptides aren't as different in weight because they're not different in size. They're just engineered in a way. They may have the same number of amino acids. So they're pretty comparable. If you administer them in a way that they're modified in the same way, I think that's key. They have the same fatty acid to enhance albumin binding and so on. This is where really differences happen there that are independent of whatever hormones in there. You can turn a mono-agonist into something very powerful with the right modifications chemically, and if you don't do that to a dual or triple agonist, then you can't compare it. But if you have the same modifications, it's the same size of molecule, you give the same dose, I think it is comparable. In terms of the high throughput we need to deliver and develop these drugs, we only looking at fat mass, at body weight, at glucose metabolism, and at food intake. That gives you a pretty good picture. I mean, the history of the last 20, 25 years shows that we are working in a field where that is pretty translatable into humans. This has been, if you look at our papers of dual and triple agonism, that looks like the clinical papers now. I guess we got really lucky, because when you look at neurodegenerative diseases, none of that's happening. So I guess it's fair to continue that way for now.
We have a question from
Torkel Johnson, University of Copenhagen. I wonder whether there might be differences in post-treatment effects, any sort of differences in hangover effects of these various drugs.
So what you're saying is when we stop treatment, what's happening with
rebound? You have the
rebound?
regain, and the very interesting point would be differences in the rate of regain, or even no regain, because you establish some kind of a permanent alterations in the regulatory systems.
Yeah, very intriguing area of research where we haven't done enough. Of course, when we stop, we then look what's happening. Everything we have seen with our drugs, there is no significant difference there, really. Which sort of makes me happy, because if we give our GIP glucagon co-agonist and we stop treatment and weight stays off, that would frighten me to death. What have we killed here? I think I still believe we need some sort of maintenance drug at some point, and having these sort of combinable tools, right, where you have a GIP glucagon and you add a GLP, you add a PPAR, you take it away, that gives you ways of continuous treatment and then adjusting for a maintenance therapy. But having a treatment that when you stop gives you additional months of relief, I don't know if I believe it or want it. That's a tricky area. We're not working on that.
There's a question up here.
This is Paul Njolster from University of Bergen. Fantastic talk and interesting data. So I was wondering, how do you envision that can be used for precision medicine? So do you think that epigenetic factors, various genetic constitutions, etc., can be important here to target different patients with different of these combinations or whatever? So one size doesn't fit all, and obviously there is a lot of differences between individuals when it comes to propensity to get obese and how fast and so forth.
Yeah. So absolutely. We know from our clinical colleagues that there are these subpopulations, right? That certain drugs work better on some people and others better on others. How to predict and find out what works with which subpopulation will require the right diagnostic tools and predictive tools. And that is not my area of research really, but at our diabetes center at Helmholtz and many national and international endeavors, ERC, etc. These things are being looked at, and so far I don't think they have delivered what we hoped they would deliver. Epigenetics and GWAS and whole genome sequencing and lots of interesting data. But in terms of the tools that tell us, based on that genetic workup, you need a triple receptor agonist. I don't think we're there yet. I haven't seen it yet. In a way, the transformation based on of clinical medicine based on these new available therapeutics hasn't been paralleled by a transformation of our diagnostics in metabolic medicine, where basically it's BMI and HbA1c, if I wanted to be facetious.
There's one more question over here.
Yeah, thanks. Jens Halling, Novo Nordisk. I'm wondering about these combination molecules with the PPAR or thyroid hormones. How confident are you that will translate to humans? Because PPARs, they give weight gain in humans, and thyroid hormone is also now marketed for MASH and does nothing to body weight in MASH patients that also often have obesity, right? In these molecules, I'm thinking you would essentially be targeting PPAR or T3 to the brain, and then also small populations in the periphery. But where would the efficacy come from then?
Well, the efficacy comes from two things. One, that you will have a concentration, a dosing of these small molecules at receptor subpopulations only that otherwise are not being selectively targeted. In addition, you have a combination of that effect with the effect of the vehicle, which in this case is GIP-GLP, dual agonist. So that is a very complex, completely different pharmacological targeting and profiling. Therefore, there's only one way to find out. We need to put it in humans.
We have a question here.
Yes, Stefan Romeo, Karolinska Institute. This is a different topic. I'm more thinking about equity. This is a huge business market we're talking about for obesity. Do you envision a way to make it more affordable?
I'm not a businessman, but I very much hope that all of these drugs are becoming more affordable every year now because of additional competition. Because of, at some point, the first ones going off-label. And I think that is an important and a good thing, because as we know, Cost of goods are relatively low or very low. So this can be produced affordably and can be offered to patients worldwide that are in dear need but can't afford it. Now, I'm saying that knowing enough about industry and drug development, that the investments that were made over the last 30 years were enormous and got us to where we are. And they need to be recuperated through the next thing. So that's how the world works. But I do hope that this is coming down. And all of you know that as well. It's difficult to understand for patients why things cost that much in the US and that much in Denmark and that much in Germany, and that feels random, and they don't understand how that is the best way forward.
Okay, Matthias, I'll ask final quick question, and then we're done. Thanks a lot for the engaging discussion. So I kind of want to take one step back. So now you took upon a new role as president of the traditional university with many different study directions. You have philosophy, you have history, you have physics. What is kind of next step for Matthias Tschöp, the scientist and the innovator in that role?
Retirement.
Okay.
Looking forward to that.
All right. With that.